Long ago, a single woman lived in Africa. She became a direct ancestor to every person alive on Earth today. She is the individual from whom we all inherit a specific piece of our genetic code. Scientists call her the mitochondrial most recent common ancestor, MTMRCA. In popular language, she's often called Mitochondrial Eve. This name can sometimes be misleading, but her story reveals a profound, unbreakable connection through the veins of humanity. Her existence is not a guess or a myth. It is a conclusion drawn from the careful study of our DNA, a biological inheritance passed down through an unbroken chain of mothers. This essay will trace the story of this shared ancestor, following the path scientists took to find her. We will journey back in time, using a timeline breadcrumb approach to mark key moments in this maternal history. The story begins with the unique nature of mitochondrial DNA and how it is passed down. From there, we will explore how tiny changes in this DNA act as markers, allowing researchers to build a vast family tree for our species. We will pinpoint the moment our shared maternal lineage began and place this ancestor in her correct historical context. To understand our connection to mitochondrial Eve, we must first look inside our own cells. Within the main body of each cell are tiny structures called mitochondria. These are often described as the powerhouses of the cell. Their primary job is to convert food into usable energy, essential for life itself. But these microscopic engines hold a secret. They contain their own small, separate set of genetic instructions. This DNA is known as mitochondrial DNA, or mtDNA. It is a small circular chromosome. It is distinct from the much larger set of chromosomes found in the cell's nucleus. Those nuclear chromosomes are inherited from both parents. When a sperm fertilizes an egg, a new life begins. The resulting embryo inherits nuclear DNA from the mother's egg and from the father's sperm. Those combine to create a unique genetic blend. However, the mitochondria, and therefore the mtDNA, are contributed almost exclusively by the mother's egg cell. The sperm's mitochondria are typically located in its tail. The sperm tail is discarded during fertilization. As a result, every child, male and female, inherits their mitochondrial DNA solely from their mother. This creates a direct, unbroken line of transmission from mother to child. Because of this unique maternal inheritance pattern, your mtDNA is a near-perfect copy of your mother's mtDNA. Hers is a copy of her mother's, and so on, generation after generation. This creates a clear, traceable line that moves backward in time exclusively through the female side of your family tree. The story of our shared maternal ancestry is written in a language of tiny, random changes. Although mitochondrial DNA is passed down as a near-perfect copy, it is not entirely immune to alteration. Over long periods, small, accidental changes can occur in the sequence of the mtDNA. These changes are known as mutations. Think of them as tiny spelling errors that happen as the DNA is copied from one generation to the next. These mutations occur at a relatively slow and predictable rate, almost like the ticking of a molecular clock. Once a mutation appears, it is locked into that maternal lineage and passed down to all future descendants. These mutations serve as invaluable historical markers, like breadcrumbs left along a trail. Imagine a mother has a unique mutation in her mtDNA. She will pass this specific marker to all of her children. Her daughters will then pass it to their children, and so on. Everyone who carries that specific mutation today can be certain that they share that one woman as a common maternal ancestor. By comparing the mtDNA sequences of people from different parts of the world, scientists can identify which mutations they share and which ones are unique to their own lineage. This allows them to group people into related maternal clans. These large maternal clans, defined by a shared set of mutations, are known as haplogroups. Within that group, a smaller subset might also share a newer mutation, forming a subclade. Just as related languages share common root words, related haplogroups share ancient mutations, letting scientists work backward to the trunk of the tree. As scientists constructed the global maternal family tree, they made a remarkable discovery. All the mtDNA lineages found in people outside of Africa belong to two major branches, haplogroup M, haplogroup N. However, within Africa, they found a much greater diversity. These African lineages were older and sat closer to the root of the tree. The oldest and most diverse branch was a massive foundational group named macrohaplogroup L. This confirmed our shared maternal line was rooted in Africa. Macrohaplogroup L is the trunk of the human maternal tree. All other haplogroups are branches from it. Looking closer, researchers found it splits into primary lineages: L0, L1, L2, L3, L4, L5, L6. The very first split at the base is between L0 and the branch L16. That pivotal fork is the most significant point in our maternal history. The woman at that junction is our mitochondrial most recent common ancestor. L0 is most common today among the Khoisan peoples of southern Africa. They carry some of the world's most ancient genetic lineages. L16 gave rise to other African lineages and to L3. L3 is the ancestor of the M and N haplogroups found outside Africa. The deepest split supports that mitochondrial Eve lived in Africa. Using the molecular clock principle, which calibrates the rate of mtDNA mutations over time, scientists have been able to estimate when mitochondrial Eve lived. The most widely accepted scientific estimates, based on a 2013 study, place her at approximately 155,000 years ago. This figure is not a precise birthday, but a statistical midpoint within a range of possibilities. It gives us a powerful frame of reference for understanding her place in the timeline of human history. This date situates her existence at a crucial period for our species. The emergence of our species, Homo sapiens, is believed to have occurred in Africa around 300,000 years ago. This means that mitochondrial Eve lived long after our species had already become anatomically modern. She was not the first woman, but was part of a thriving population of modern humans who had existed for well over 100,000 years before she was born. Her significance comes not from being the first, but from being the one whose maternal lineage was uniquely successful in its survival. The next major event on the timeline is the Great Out-of-Africa Migration. This is when small groups of modern humans began to leave the African continent. They spread across the globe, eventually populating Asia, Europe, Australia, the Americas. The main wave is dated to around 60 to 70,000 years ago, roughly 90,000 years after mitochondrial Eve. During that long interval, her descendants lived and diversified entirely within Africa, giving rise to the various L haplogroups seen there today. The name mitochondrial Eve often creates confusion, conjuring images of a biblical Eve who was the only woman on Earth. This is perhaps the most significant misconception to clarify. Mitochondrial Eve was not the only woman alive during her time. She coexisted with thousands of other women in other human groups across Africa. Many of these other women also had children and contributed to the nuclear DNA of people living today. They are our ancestors just as much as she is, but their direct matrilineal lines have since died out. A maternal line becomes extinct whenever a woman has no daughters. If a woman has only sons, her mtDNA line stops with her, as sons do not pass it on. If she has no children at all, her line also ends. Over thousands of generations, through random chance alone, every maternal lineage that was contemporary to mitochondrial Eve eventually came to an end. Hers is simply the only one that, by luck, managed to produce an unbroken chain of daughters leading all the way to the present day. Another misunderstanding is that she was special or superior. There is no evidence she was stronger, smarter, or more fit. Her lineage's success was a matter of statistics, not destiny. Finally, the title is relative. The MTMRCA is simply the most recent woman from whom all currently living people descend through a direct maternal line. The story of mitochondrial Eve is ultimately a story of profound connection. It is a scientific confirmation that every one of the 8 billion people on this planet today shares a common thread of ancestry. This thread, woven through the mitochondrial DNA inside our cells, ties us all to a single woman who lived in Africa over 150,000 years ago. No matter our nationality, ethnicity, culture. We are all distant cousins, united by this unbroken chain of mothers. This knowledge does not erase the beautiful diversity of human cultures, but it does place it upon a foundation of shared biological heritage. Her legacy reminds us that our human story is one of migration, survival, continuity. The journey of her mtDNA from a single woman in Africa to every continent on Earth is a testament to the resilience and adaptability of our species. The mutations that accumulated along the way tell a story of ancient journeys, of populations splitting and moving into new lands, and of adapting to new environments. Each haplogroup is a chapter in this grand narrative, and every person's mtDNA is a personal copy of this incredible history book, connecting their own life to the deep past. Science is constantly refining our understanding of this story. As new genetic data is collected from populations around the world and new techniques for analyzing ancient DNA are developed, the picture of our past becomes clearer. The estimated date for mitochondrial Eve may shift, and our knowledge of her descendants' journeys will grow more detailed. But the core truth remains unchanged. The human family has a single, traceable maternal root.